Good afternoon, everyone. So welcome back to the chapter 17 live. So actually we already 2 weeks. This live I think 2 weeks. 1 week only lah. 1 weeks. This live I already assist 1 week. So in this 1 week I already touched about the chapter 17. So finally we achieved the last topic of this chapter. That is topic mutation. Okay, so mutation, but I will proceed to part 1 and part 2 because mutation topic is also quite long topic. So I will proceed to part 1 and part 2, separate into part 1 to part 2. So for this topic, I will also consider part 1. So part 1 topic, I only will explain the gene mutation. So for part 2, I will explain chromosomal mutation. This is for part 2. This I will proceed to the live at on 5 p.m. lah. For this slide, I only will proceed to the gene mutation one. Okay. So firstly, you have to know what is the meaning of mutation. So actually, you guys have experience syllabus, you already know. So I just repeat lah. Maybe revise back ah. Some maybe already forget, but I just want to remind that. So it is the abnormal change in the structure or content of genetic material leading to structural and functional change of protein product on the individual. So you have to know that our gene sequence in the DNA will be our genetic information in DNA actually will be transcribed to mRNA, and then it will transcribe to protein also, right? So, if mutation occur, it means that the abnormal change the DNA sequence will cause abnormal change. Means the DNA sequence is already changed. So since the DNA sequence is already changed, mRNA sequence will change. And mRNA change, protein structure also can change. So maybe the protein is cannot form, or maybe the protein is formed but cannot function properly. I don't know. We need to see what kind of mutation occur in the DNA. So the mutation is very can say that it is very serious problem lah. It can cause very serious problem for the human health, for the organism one. Because if the protein of the organism cannot function properly, normally this organism may have a serious disease or serious effect to the organism. Okay, so for this live, I will more focus on gene mutation. So gene mutation means that it is the change in the sequence of nucleotide based of DNA to particular gene. Maksudnya gene mutation is the gene awak tengok is the DNA, right? DNA is composed of many nucleotide, right? Like G, C, T, A, T, T like this. So this means that gene mutation is maybe some of this nucleotide is already changed. Sequence is already changed. Maybe some this nucleotide, maybe the A is changed to T, or this T maybe changed to C like that lah. This is because as gene mutation. So it is it is simply called gene. So it is the gene sequence. The sequence of the gene is changed. Okay. So there are 4 types of gene mutation you have to learn. First one is substitution mutation. Second one is insertion mutation. Third one is deletion mutation. And the final one is inversion mutation. So apa mutation is this? Okay. So for the first one, substitution mutation. So substitute. What is mean by substitute? Pergantian. If you do the maths, if you do maths, you always know, right? Substitution. If you want to solve simultaneous equation, one of the method is substitution method. So it is just same saja. Maksudnya pun sama. So substitution is the substitution of nitrogenous base in a gene. Maksudnya, if your one of the example for the disease in substitution mutation is sickle cell anemia. Okay, for sickle cell anemia, the normal DNA is CCT, GAG, GAG. Ini normal punya. But and then it will produce normal mRNA lah, CGGA, GUC, and GUC lah. GUC lah, CUC lah, and then this is the amino acid will be produced. I think yang ni salah aku. CUC actually is glutamine. Sorry. Yang ni. It should be same. Yang ni pun sama. Salah. Sorry, salah. Maybe salah lah. Glutamine. Maybe you can refer to your Oxford textbook. Okay. Okay. So this is the normal DNA. But in mutated DNA, awak boleh tengok yang saya highlight punya. For the second base pair, awak tengok sini. Second base pair, the normal A is changed to substitute by T. Dia sudah tukar kepada T lah. So this also will cause the mRNA also change. So dia sudah kepada T, so maksudnya complementary to T is A. So the mRNA produced is CAC lah. So the CAC, the amino acid is actually valine. It will cause valine. Bukan lagi glutamin lah. Sepatutnya adalah glutamin. In the amino acid, for the amino acid produced should be glutamin. But since the nucleotide already changed, so it already changed to valine. So totally not same. The amino acid is already totally different already. So this is because as substitution mutation. The substitution mutation is one of the nucleotide base. It changed to substituted by another type of base. Don't know. Maybe A, it can change to T, C, or G. Something like this. So this is substitution mutation. Another one is injection mutation. So injection mutation, insert, what is mean by insert? Kami tambah. Sepatutnya dia hanya sepatutnya dia macam ni, tapi kami tambah satu lagi nucleotide base in a gene. So like this. This is I use mRNA lah. Normally I use mRNA. Actually the mutated is DNA one. But I just want to mention mRNA lebih senang sikit lah. Normal mRNA produced should be AA. The normal one, if you see the tengah-tengah punya, awak boleh tengok ni. You see this part. So actually the normal one is AAGUUU. So AAG will produce translate to become lysine, and UUU will translate become phenylalanine. Phenylalanine. PHE is phenylalanine. I'm not mistaken lah. Phenylalanine. And mutated mRNA for injection mutation, maksudnya dia tambah satu C dekat tengah-tengah ni. So dia tambah satu C dekat between the A and GD. So dia akan jadi AAC lah. Lepas tu GUU. Why? Because you have to know genetic code is with as triplet base. So kalau macam ni, semua genetic code, the reading of the genetic code will be shift to the right one. All of this will shift to the right. So the new C is added, so it becomes AAC, and the G will become another base pair, another type of genetic code. C will be insert into it, and then the G will be shift to the right and will read as another type of genetic code. And you can see that if you like this, all of this yang belakang punya semua ni, semua the amino acid sequence will also change totally. Langsung tak sama. Kecuali yang ini sahaja. Hanya methionine ni sama sahaja. Yang lain semua, if let's say there are maybe I have 100 polynucleotide. So if this happen, hanya satu nucleotide remain the same. Yang 99 semua amino acid, 99 amino acid totally different at all. Dia langsung tak sama. So means that this will produce another type of protein lah. Bukan yang protein yang normal lah. Another type of protein I also don't know lah. So you can see that if G is added, AAC dia sudah jadi ASN. ASN already asparagine, if I am not mistaken. Asparagine. I already forget asparagine, I think. And GUE is valine. Dia sudah tukar lah. Bukan sahaja ini, yang lain pun semua tukar. Semua amino acid akan tukar juga. So if you see that lysine and phenylalanine dia sudah tukar kepada asparagine and also valine. Langsung tak sama. And also and so on. So injection mutation is very very serious mutation because it can cause serious effect for onward after the mutation start, after the mutation. So after the downstream after the mutation, the all of the amino acid sequence is totally changed. But injection mutation actually not only add one nucleotide, actually it can directly add 3 nucleotide also. If it is added directly 3 nucleotide, like nucleotide, normally it will not give serious effect. Normally it will not give serious effect. Normally. But if only add 1 or 2 or 4 or 5 like that, not 3, other than 3, 1, 2, 4, 5, 1, 2, 4, 5, 1, 2, 4, 5 like that, it will give very serious effect. Okay. Next is deletion mutation. So delete, what is mean by delete? Kami hapuskan salah satu nucleotide. So original nucleotide base has been deleted. So for like this, you see this AAG. AAG actually it should be AAG and UUU and will produce lysine and phenylalanine lah. But because of this A, dia sudah delete lah. Kami delete. Ini A ni kami hapuskan. Kami tak mau ni A. Because of deletion mutation. So, it will become AGU. And then, this one, you will see that it only have 2 nucleotide base lah. Actually, not only 2 lah. Because actually, MIS will and other type of the genetic code will be best to it. That's why I just want you know. So means that yang U ni actually is U. The first U dia sudah pergi ke depan lah. Akan combine ke sini form another type of genetic code. And then this one yang lain yang maybe this one have ACG. Lepas tu A ni dia pun akan substitute ke sini become UUA like that lah. No more genetic code pun akan change one. So it is have also the serious effect same as the injection. So like this, if you see methionine is same but totally different lah second one. Lysine dia sudah tukar kepada serine and then this one no amino acid at all. But actually not lah. Actually got amino acid one but the amino acid onward also will change. Dia bukan jadi phenylalanine lah. Dia akan jadi amino acid yang lain lah. The amino acid will also change at all. So means that the protein sequence will also change. And then will cause the protein cannot function normally lah. So deletion mutation is also can cause very serious effect to the organism. So the final type mutation is inversion mutation. So inversion mutation is nucleotide base are inverted each other in a gene. So what is mean by invert? Bahasa Melayu terbalik lah. Kami terbalik dia. So maksudnya if you see this, it should be ACG CUA. Lepas tu AGG. But if you see that the G and C here, they are terbalik. We invert this C and G. So dia tukar kepada AGC and GUA. Awak tengok tak? C, G, dia sudah terbalik. So if you terbalikkan dia, amino acid sequence pun tak sama. So but for this, it is more does not give extreme serious effect lah because only these 2 amino acid are changed only. Compared to the deletion and injection, all of the amino acid sequence are totally changed after the mutation occur. But for inversion, hanya these 2 amino acid, the inversion occur at that genetic code will cause the change of amino acid sequence only. Yang lain semua remain sama. So only these 2 amino acid is changed. So especially it is arginine, leucine. But sekarang dia sudah tukar kepada serine and valine. So total ada dua ni adalah sama, tapi ya onward punya semua adalah sama. Okay, so this is the four main types of the gene mutation. Okay, next, this is the new things. It is the effect of the mutation. Tadi adalah four types. Ini adalah empat jenis mutation. This is the effect of the mutation. These four type of mutation can cause these four effect. So there are total four effect mutation you have to know. First one, friendship mutation, silent mutation, mid-sense mutation, and non-sense mutation. Okay, what is this meaning? Friendship mutation. So, friendship mutation means that the number of nucleotide, it is the effect of mutation where the number of nucleotide are added or deleted that are not in number of 3. So, injection and deletion, and it will cause serious effect because it is not under E3. So if you see the effect, what are the effect? So it will provide extreme harmful effect to the protein structure, a very harmful effect. And because all of the amino sequence are changed after mutation. So same like the injection and deletion, I already say, if not 3, kalau bukan 3 base pair, bukan 3 3 base pair ni, at the 3 base pair, insert or delete, and ini delete, hanya 1 atau 2 atau 4 yang delete, it will cause all of the amino acid sequence will affect. So all of the amino acid sequence are not same already. So this will cause the protein will be denatured. It already denatured one. Because the protein shape is totally cannot form. Connect form, and it will be denatured and totally unfunctional lah. Tak boleh function lah. Tak ada protein lah. The protein is absent already. No protein at all in this organism. So friendship mutation is very it is effect of the mutation is very give a serious effect one. So for normally if the people get this friendship mutation effect, normally the people have the highly serious disease lah will get. Okay, next is silent mutation. This effect actually is occur but do not change any DNA sequence. Does not change any DNA sequence. Masuk apa? Masuk masuknya adalah what change the DNA sequence tapi the genetic code is still encoded with same amino acid. So if you know that one amino acid actually can encoded by many type of genetic code, right? Dia ada banyak genetic code can coded with this one amino acid. So example, kalau if you coded with like I don't know because I don't memorize the genetic code table. So maybe this one I known as AAA lah, AAC, AAU dan juga AAG. Yang ni empat ni coded with same amino acid, but actually the normal one is AAA. But now because of mutation, but this A I changed to AAC. So actually it is cause it will result in silent mutation because no any harmful effect at all. Because this sequence also was coded with same amino acid. So this is why that does not provide any harmful effect. Dia tak bagi any harmful effect because the amino acid is still same. The protein structure is still same. Remain unchanged. So the protein can function normally. Right? So this is we call silent mutation. Although ada mutation, tapi ini mutation tak ada effect langsung because the amino acid coded is still same. Next is about missense mutation. This mutation is the change in the base sequence alter a codon so that one amino acid in the protein is replaced with a different amino acid. Yang ni macam substitution lah. Hanya satu amino acid is changed, like substitution and inversion, like this. One of the codon is changed, so actually it should be arginine, But now it changed to serine. One of the amino acid sequence is changed already. Because the replaced with a different amino acid. So this one is caused by substitution. Okay, and then this will provide some harmful effect, not too much, just some saja, compared to this, compared to French mutation, because French mutation is totally no protein at all. This one, maybe it can produce protein, but the protein structure maybe is different. It not different, not maybe, it must be different, but the protein still have the shape, the similar shape from the normal one. But just that the protein cannot function properly lah. Yeah, it cannot function normally lah. But it still can function. Compared to French mutation, French mutation is totally double function already. Means then still can function, but cannot function properly. Because one of the amino acid is already changed to replace to another type. Okay, next is nonsense mutation. This one is actually also a serious effect also, because if the sequence of base is changed to the stop codon, maksudnya salah satu dari tiga ni, UAG, UAA, UGA, kalau dia tukar kepada sini, means that it is stop codon, so that the translation will totally stop. So normally this one will occur in substitution substitution and inversion. That's inversion also. But actually insertion and deletion also have the possibility to happen, but normally will happen in substitution and inversion. Okay, so this part, however, this may provide serious effect. Does not necessarily it will provide serious effect, because it need to see that the mutation at back. So that if the mutation you see, kalau mutation dia dari sini. So I assume that this is normal start codon. This is normal stop codon. However, the first case is the mutation occur at here. Occur at here and cause the nonsense mutation. And then so it stop at here already. So I use different color. Sikit. Okay, ada satu mutation at here, and then this will cause it already stop. So this one actually does not give very serious effect also, because the amino acid still can produce until here. Just that, and some of the amino, maybe 3 or 4 amino acid is absent lah. So it still can form protein. Boleh juga, sometimes boleh juga, just sikit sahaja boleh je. Tapi functionnya maybe must will have the abnormality lah. But if let's say the mutation is occur at here, and then dia stop lah. Oh, ini terlampau lah. Because the amino acid sangat kurang. Hanya maybe from the start codon until here hanya 4 amino acid saja. Macam mana dia boleh menghasilkan protein 4 amino acid? How to form protein? Actually, one polypeptide chain need at least 100 amino acid, right? Hanya 4 amino acid, macam mana dia boleh form protein? Langsung tak boleh. So this one will provide serious effect compared to this one. So this one will give not too serious effect just provide some harmful effect. This one, if the mutation occur here, it must provide serious effect. Why? Because the amino acid, the polypeptide chain is too short. So this is why that if the nonsense mutation occur, the polypeptide will be shorter than expected due to the stop codon. Compared to the mean sense mutation, yang ni, mean sense mutation, the polypeptide length is still same. For example, kalau normal ni, it will produce 100 amino acid to form one polynucleotide. Mean sense mutation also will produce 100 amino acid. But nonsense takkan mesti kurang daripada 100. Mesti less than 100. Compared to silent totally same 100, but friendship need to see lah. Either it is injection or deletion. Kalau injection, it will must more than 100. If deletion, it will less than 100. See lah, compare lah. You have to see lah. Injection more than 100, deletion less than 100. But for mid-send and non-send is totally same. Mid-send and non-send, mid-send is still same 100, but non-send it will less than 100. So this is why that the polypeptide chain will be shorter. Yang paling penting dia akan jadi pendek. Kerana stop codon ada. So tak ada, so means that the protein structure is unstable and then may result is no protein to the cell. If the stop codon is the polypeptide chain is too short, it is unable to form protein, so actually that cell is also not have protein. Kalau tapi if the cell is not too short, actually it still have a protein, but the function of the protein is not good lah. Have the immobility lah. So these are the 4 effect of the mutation. Yang ni paling penting lah. Because this one is new to you and normally in essay question dia suka panggil kamu buat perbezaan punya. Either dia cakap perbezaan mean sense until non-mean sense or friendship until silent or either silent dia bisa tak tahu lah. Macam-macam lah. Normally MPN suka soalan macam ni lah. Differences. Non-mean sense, unsend, silent, and friendship or either silent mean sense, I don't know. So you have to know detail lah. Yang ni 4 jenis ini SPL awal sudah belajar. So normally MPN tak fokus sangat. Yang fokusnya dia akan fokus ke sini. So you have to know. So this is not the type. Awak don't. If the question structure question want to mention what is this mutation, normally you have to mention the one. This is the effect. It is not the type. Dia bukan jenis. It is the effect of the mutation. Okay, so hopefully this live is end. So this topic, gene mutation topic is senang lah. Tak susah sangat. So, okay. Same as the same. You can scan this feedback form, and if you like my video, you can subscribe my channel so that you can take the latest information and also join my channel, also Telegram channel, and also my Facebook. Okay, thank you. Have a nice day also, and see you on 5 p.m.